Welcome, students. This is my fourth lecture for fifth semester students in Unit 1 for Pharmacognosy and Phytochemistry 2. In which today we are going to discuss about the how to investigate the biosynthetic pathways. We have discussed about the biosynthetic pathway in the last lectures regarding succinic acid pathway and also acetate pathway, which has many other option of asking like HMG-CoA pathway, mevalonic acid pathway, terpenoid pathway, and isoprenoid pathway. So they can ask in this way because all of them are coming inside as a product in the pathway and a precursor, you can say, intermediate product, which can give rise to various secondary metabolites. So we have understood about what is a primary metabolite and what is a secondary metabolite and how these biosynthetic pathways are formed or made the transformation using various enzymes for the development of new structures like alkaloid, glycosides, steroids and hormones. These all of them and amino acids, basically they are all an precursor intermediate product for developing various secondary metabolite. How to find out this biosynthetic pathway? How to investigate this biosynthetic pathways? So that's we have isotopes, radioisotopes, which can be utilized by means of producing certain compounds. What is the intermediate product which we know about the amino acids, which can develop or a labeled compound can be prepared. Labeled means incorporating these radioactive isotopes. And we can make various tracer technique, tracing. What do you mean by tracer? Tracing? Tracing means identification, inventing, investigation, tracing the compound, tracing the change, the biotransformation and the product. What is the product formed from the basic primary metabolites of like what we studied in the pathways like pyruvate, glucose and acetyl-CoA. These all are the starting product for various biosynthetic pathways. An intermediate product, we know that like succinic acid, we know succinic acid, quinic acid, corosmic acid, and we know various intermediate products. And even acetate pathway, we know mevalonic acid, HMG-CoA, geranyl pyrophosphate. Why not? We know about the 5-membered carbon atom compounds, isoprenoid units. So they are all some of the intermediate products, precursor, which can be prepared with the radioisotopes incorporating like carbon, hydrogen, whichever present in that particular structure can be developed into an isotopes. So we should know about how to prepare this labeled compound. Definition, especially before going to the topic, I would like to tell about certain things in a brief manner. What's the inside these topics? If you know that, if you listen to the class and if you write down once, you can remember. I'm damn sure it's not necessary for you to read it again and again. You can recall what you have brain have understood from this topic. That is the way of studying. Instead of reading again and again, recalling what you have understood is very interesting for making your brain to work and to memory and to write the exam. Tracer techniques, here use of and also other techniques in the tracer techniques, using of isolated organ, grafting methods, and the use of mutant mutant strains from microorganisms or from enzymes. These all we are going to discuss today. And before going to the topic, we should know about tracer techniques. Tracing, what is its definition and all it is needed. Living plants considered as biosynthetic laboratory. Our even animals or the human being, it's also a biosynthetic laboratory where the primary metabolites and the primary as well as the secondary metabolites. We know that secondary metabolites are produced or formed or biotransformed from primary metabolite by means of various steps using various enzymes. And different biosynthetic pathway what we studied, just I am giving you succinic acid pathway for aromatic compounds, and mevalonic acid pathway, acetate pathway, HMG-CoA pathway, isoprene pathway, terpenoid pathway. So you see, all of them are, if you study one pathway, how a cholesterol is formed, how an terpenoid is formed, it is possible for you to make a starting point from acetyl-CoA. So the interesting to know, just making into divide only two pathways, two divisions. The students can easily study. I have given all the previous lecture. If you listen, you will understand it's not a big issue to study about these biosynthetic pathways. Various intermediate steps, intermediate products involved in biosynthetic pathways in plants can be investigated by means of the following techniques. That is the tracer technique. So here, first tracer technique, we have some different methods, branches, any techniques, difference will be there. How to trace them. Number one, precursor product sequence method. And number two, double or multiple labeling method. Number three, competitive feeding method. Number four, isotope incorporation method. And number five, sequential analysis. So these all techniques can be followed. Apart from that, other techniques using of isolated organ, even in animal and in the plant also. Grafting methods, which we commonly do, that is also one type of identification of what happens to my precursors and which one is the responsible, which part is responsible for production of this secondary metabolite. We know that in plants sometimes some secondary metabolite are present in leaf, some of them are in bark, some of them are in root, some of them are in flower. So parts are responsible for giving you the medicinal secondary metabolite for you to have in medicinal use. So use of mutant strains also we are going to study. Some organisms are used, some microorganisms. Some of the enzymes are also used for the study of how to trace them. What's the change of certain secondary metabolites takes place, can be identified. So these are the techniques which have to explain. If they ask 10-mark question, very detailedly it is possible. Definition, when you want to define, it can be defined as a technique which utilizes a labeled compound. Now the question compounds, what is the labeled compound? In the beginning itself I told, radioisotope compound like carbon, nitrogen, sodium, potassium. There are iodine which has radioisotopes which are labeled compound, means it will be incorporated which will be added to the normal carbon atom will be converted into carbon 14 if there is carbon 13. Carbon 12 is the normal without radioactive isotopes. So we will have a discussion what is isotopes, what are radioactive isotopes, we should know an idea. That also I have added in my lecture in the beginning. To find out or to trace the different intermediate and various steps in biosynthetic pathways in plants at a given rate and time. So kinetics, the rate of reaction, what's the time, and product, what's the quantity of the product, rate and the time can be identified. Kinetics also gives detail. Or this can be defined as, in this technique, in this tracer technique, in this investigation of biosynthetic pathway, different isotopes, mainly the radioactive isotopes, which are incorporated into presumed precursor. Precursors are which will give the particular product, which are the intermediate or the responsible, which are sometimes will rate limiting step we used to say. Precursors of the plant materials, or you can say markers, are used as markers in biosynthetic experiments. So they are called as the investigation. How to investigate a biosynthetic pathway? We studied biosynthetic pathway and we should know how they are investigated. How they are identified by the scientist is also needed for a pharmacist should know that before going to a research. You see, this is a picture to show you a compound having an intermediate amino acid, isoprenoid unit, nitrogen, carbon. You see, this nitrogen can be made into an radioisotope compound. You can see that is N15. N14 is normal, N15 is radioisotope. Carbon 13 is a radioisotope. You can see here in the picture, down the picture I have given that. What is its MEZ? What is its molecular number? About the proton and the neutron numbers will give you the mass. So the mass changes according to the number. So you can see that nitrogens which is present in amino acids can be changed into radioactive isotope nitrogen, that is called labeled compound. And when we can see that, what is the product, what is the change happening to this radioisotope element? You can see this aromatic carbon atoms can also be taken into consideration, made into carbon 13 as a radioisotope element. So the labeled compound can be prepared by use of two types of isotopes. How you are going to label the compound preparation? There are radioactive isotopes, stable isotopes. Radioactive isotopes are H1, carbon 14, sodium 24, potassium 42, sulfur 35, phosphorus 35, iodine 131, delay in emission of radiation. They have the fluorescence and the emission. You know about the radioactive isotopes can emit the radiations. The radiations of gamma, beta or gamma rays can be identified by various instruments, and when you are incorporating in a precursor, when it is injected or it can be fed to the plant or an animal, how you are diagnostic things happening, doing in a clinical side, identifying that radioactive isotope compound, what is the change has happened, and we can find out what is the biotransformation taken place. This is the main identification done in the tracer technique using a labeled compound. Now we are going to talk about the isotopes. What is the isotopes? We should know that. You see, normal carbon atom 12, the atomic number, which shows that 6 proton and 6 neutrons. And carbon 13, which is a radioactive isotope, which has 7 protons and 6 neutrons. And we can see in carbon 14, it has 8 protons and 6 neutrons. You can see the sodium ions. Sodium isotopes, same atomic number, different mass number. So you can see that 23, it has 11 protons and 12 neutrons. And 22, 11 protons and 11 neutrons. And even you can see 13 neutrons containing sodium 24, radioactive isotope. In hydrogen, you can see the hydrogen 1, which has mass number 1, only 1 proton. When having 1 proton and 1 neutron, the mass number becomes 2, that is hydrogen 2, which is deuterium. And when mass number 3, which has 1 proton and 2 neutrons, they are tritium. We know that this is the electronic structure of presence of proton, neutron and circulation of electrons, which has an stable stage and an excited stage for any atoms and an electrons, which we have studied about the quantum number and hybridizations. All these things you have studied. The basic physical chemistry is important for the student to revise and to know about what do you mean by isotopes, what do you mean by labeled compound. If you listen to the class, if you listen to the very clearly and write down, and it will not go away from your brain. It will be easy for you to remember. For biological investigation, carbon and hydrogen are usually used. For metabolic study, you can use sulfur, phosphorus and alkali and alkaline earth metals are used. For studies on protein, alkaloids and amino acid, labeled nitrogen atom given more specific information, as amino acids always contain nitrogen. H3 tritium compound is commercially available, and stable isotopes like H2, carbon 13, nitrogen 15, oxygen 18 are used. If you see this picture about nitrogen 14, usually they have 7 protons and 7 neutrons and 7 electrons. And when you see that stable isotope of nitrogen 15, you can have, it has 7 protons, 8 neutrons and 7 electrons. And when we come to 16, nitrogen 16, it has 7 protons, 9 neutrons and 7 electrons. Used for labeling compounds as possible intermediates in biosynthetic pathways. Usually methods of detecting are, even you are using mass spectrometry or mass spectroscopy, you can say, or mass spectrometry. Here we can use the nitrogen 15 can be identified, oxygen 18, the isotopes, stable isotopes can be identified. When you are labeled compound using in a biosynthetic study, you can find out how it is changed and where this element have been transferred, what is the form of the structure have been changed. NMR spectroscopy will give you detail about the H2 deuterium and the carbon 13 radioactive isotope, which can also identify where it is present in structure, how it is changed. If suppose you have labeled the phenylalanine, and what is the change have been happened in the phenylalanine when you are giving to the living plant, and if you fed the plant in the root or a shoot or in a leaf or a flower, how it is changed after a certain period of time. The particular compound can be isolated and can be identified by using in which type of structure this radioactive isotope is present. If the phenylalanine is converted into tyrosine, you can say it is converted into tyrosine. If it is converted into another compound like any other molecules which has an aromatic compound, then we can say this is responsible, phenylalanine is responsible for converting to become this type of compound. So this is a basic instruments and identification. The labeling of compound plays a major role. And we should know what is the significance of tracer technique initially. Tracing of or identifying of a finding of a biosynthetic pathway. Example, by incorporating the radioisotopes of carbon-14, that is radioisotope carbon-14 into phenylalanine, the biosynthetic cyanogenetic glycoside pronacin can be detected. Location and quality of the compound of a compound containing trizo carbon-14 labeled glucose is used for determining of glucose in biological system. Different traces for different studies. For studies of nitrogen and amino acid, labeled nitrogen gives specific information than carbon. It's convenient and suitable technique. Criteria for tracer technique, we should have some criteria which should be followed. The starting concentration of tracer which you are using must be sufficient to withstand the resistance with dilution in the course of metabolism. We are going to feed the particular tracer, particular labeled compound to the living organism. So it should withstand what concentration which you are using it. You cannot use high concentration also. We cannot use very diluted which is not having any exposure towards the reaction. So a normal concentration should be identified. We should have the reference and trial and error by that which is possible to find out. We should have a resistance with the dilution of the course of metabolism. And proper labeling is very important. For proper labeling, physical and chemical nature of the compound must be known. Suppose if you are labeling carbon-14 with phenylalanine, we should have to find out proper physical and chemical nature of the phenylalanine with carbon-14. If you are taking glutamine or glutamic acid with nitrogen, 15 is incorporated or it is labeled with nitrogen 15. We should proper labeling. We should find out whether it is having the physical and chemical nature of that particular compound, that is glutamine, nitrogen with 15 labeled must be known very clearly. What is its stability? What is its proper labeling? We should identify. Example for glucose 13. C6 means 6 carbon atom present in the glucose is completely labeled with carbon 13. You see the formula 13 is because carbon usually 12. But you are making a radioactive isotope carbon to a glucose, changing that carbon atom and labeling, that is called labeling. This is glucose molecule with 6 carbon atom. All the carbon atom is made into carbon 13. See that identification which I have shown by rounding it. 1, 2, 3, 4, 5, 6. 6 carbon atom have been labeled with carbon 13. This is that labeling of a glucose molecule. Simply, a student should know what is labeling. What is radioisotopes? How you are labeling a compound and it is fed to the particular living organism. plant or animal and finding out what is its biotransformed product. That's tracer technique. That's it. Once if you understand or once you understood this basic idea, what I am saying, will give you the clear cut of picture. That's all. Once if you know that, labeling of compound with radioactive isotope, carbon, nitrogen or sodium or potassium or sulfur or iodine, whatever may be. It should be made into a labeled compound in a precursor or intermediate product which has to be fed to the plant or animal, and studying about the biotransformation, how the pathways can be studied. The labeled compound should involve in the synthesis reaction. You cannot inject any product and find out what is its change. We should know that what is the main chemical present in that. We should know that. And we are studying about the pathway. Once you know that what's the compound present in the particular plant or the animal, then only it is possible for you to find out or possible for you to make the labeled compound to be fed into that particular organism. You see, this is an example of L-glutamine, nitrogen, 15 nitrogen, that is 15 atomic number having nitrogen, an isotope of N, 2 molecules have been added. We know that this is glutamine having nitrogen, and this nitrogen is made into an isotopes N15. See that I just rounded and showed. This will become a labeled compound which can be fed into to find out what happens to this glutamine. How this glutamine is giving rise to another compound, another secondary metabolite. Labeled compound or labeled should not damage the system in which it is used. Sometimes if you are using high concentration, they may damage the living organism or living metabolic pathways. So the study will be interrupted. So these all the things, main criteria for making an experiment with tracer technique. L-phenylalanine, another example, I was telling about phenylalanine. We have studied in phenylalanine. So the students should know about what is the basic structure. As I told in the class, structures should have an imaginary picture should come in your side, and then you have to write in the paper. Not seeing, reading again and again. You have to recall how to write a chemical structure. You see, this is the phenylalanine, C6H5CH2CH, and nitrogen is 15 atomic number nitrogen with H2, NH2, CO2H. See the structure. How the nitrogen is made into 15 nitrogen isotope. And pyruvic acid, 13 carbon atom is present in this pyruvic acid, CH3CO, and this carbon atom is made into an isotope, radioisotopes C13O2H. See the structure. How that carbon atom is made into an isotope carbon atom, and it becomes a labeled compound. So this is the example of telling out to the student should know, because you have come across this pyruvic acid and acetyl-CoA and phenylalanine. These all are precursor, intermediate product, and you can see the example of phenylalanine labeled with nitrogen 15, pyruvic acid labeled with carbon 13, and acetyl-CoA labeled with carbon 13. Acetyl-CoA is always coenzyme A, it's having phosphate and erythrose and even urea. So you can see adenine, not urea, purine derivative. So the purines will be connected as an enzyme, but only it is having 2 carbon atom compound. That is why acetyl-CoA we have come across many times. The structure looks very big because phosphate and purine derivatives with phosphatase. Advantages of this tracer technique: high sensitivity, applicable to all living organisms, and a wide range of isotopes are available, and a more reliable, easily administering and isolation procedure gives accurate result if proper metabolic time and techniques applied. Limitations are also there for this experiment. Kinetic will affect this our experiment, that is time and the product obtained. Chemical effect will be there when you increase the concentration or reduce the concentration, or sometimes what the chemical which you are using, labeling difficult, all this thing, chemical effect will be there, radiation effect will be there, and radiochemical purity is very it will affect, actually it is also having an influence of affecting your experiment. High concentration distorting the result, that is also which is very important to know. And how to make this labeled compound. Now you have an idea about labeled compound, you have an idea about isotopes, you have an idea of the mass number, you have an idea about this proton, neutron and electron. Now you know how to make the labeled compound. How to make? You can make a labeled compound by growing chlorella. That means growing the plant or the organism in the atmosphere of the particular radioactive isotope. And catalytic exchange, sometimes you can use catalytic exchange, is also possible. Then organic synthesis by using Grignard reagent or certain reagents with radioisotope compounds can also make a labeled compound. So how to make a labeled compound? We can, three types of things have to be remembered. You can, growing chlorella and a catalytic exchange, organic synthesis. These three techniques can be used. So the labeled compound produced by growing chlorella in atmosphere of 14 carbon dioxide. 14 carbon dioxide means the carbon CO2. The C is not C12, it is C14. All carbon atom will become carbon 14 labeled. And you can see tritium labeled compound is commercially available. How the tritium labeling is affected by catalytic exchange in aqueous media by hydrogenation of unsaturated compound with tritium gas. Is possible for you to get 3H compound having. And tritium is a pure beta emitter. It's a pure beta emitting radiation. Low insensitivity and its radiation energy is lower than carbon 14 isomer or isotope. By the use of organic synthesis, you can use Grignard reagent CH3 MgBr with reacting with carbon dioxide, the carbon dioxide which having the carbon 14 isotope, which gives CH3 acetic acid with carbon 14. Acetic acid CH3COOH. That carbon is also getting the isotope of C14. When you add a Grignard reagent, that can give an acetic acid CH3C14COOH and MgOHBr. This can be obtained. So three things, how to make a labeled compound. A student can write what is a labeled compound, what is isotopes, what is radioisotope, what is stable isotopes, and how can prepare a labeled compound. Finished. So basically what is labeled compound, what is its application, N-tracer technique, how to prepare a labeled compound is very important for the student should know. So there are three steps, three methods: growing chlorella, catalytic exchange, and organic synthesis can be applied, and other techniques can also be used, but at least you should know these three. Precautions, what are the precautions which you have to carry out when you are doing this experiment, investigation of biosynthetic pathway? The precursors should react, the precursor which you are using, the precursor which you are selecting for labeled compound or labeling a labeled precursor, they should react at necessary sites of synthesis in the plant. Otherwise there is no use. The plant at the experiment time should synthesize, when you are doing the experiment, the plant should have the ability to synthesize the compound under investigation. So see, sometimes the infant plant will not synthesize the secondary metabolite. It needs a developed plant. So that, and a plant or a developing stage of the fruit or developing stage of the flower. So you should know the which time the particular secondary metabolite which you are going to study. So the very important point, the valuable point, plant at the experiment time should synthesize the compound under investigation. The dose given or the fed to the plant or the animal or the living organism is for short period. Introduction of labeled compound. How can you feed for your experiment in a plant? Root feeding can be done, stem feeding can be done, direct injection to the plant can be done, infiltration can be done, floating method can be done, and spraying techniques. I am not giving all the details about this, how they are doing. Just you can have some reference or definition, how to feed all these things. Because we are not going to have a full detail how the method is followed. Just a student should know what is a tracer technique, what do you mean by biosynthetic investigation, how the labeled compound is used, and what is labeled compound, what is the labeling is done by isotopes, what type of isotopes, why they are called as isotopes, what do you mean by radioactive isotopes, how we are going to identify this radioactive isotopes, how they are labeled compound conversion, and how it is made into another structure can be identified. We have already explained about NMR and mass spectrometry. That also we should know about that instruments, and some of the instruments basically which is used for identifying this radioactive isotopes also important. Separation and detection of the compound can be done by means of this Geiger-Müller counter, liquid that is scintillation counter, gas ionization chamber, Bernstein counter, mass spectrometry, NMR spectrometry, autoradiography, radio paper chromatography. So you see Geiger-Müller counter. It's the picture which is given, an outline picture which is digital, having anode and a cathode with an electric leads which contain an ionizing gas atom. Ionizing radiation takes place due to an organ gas which we can produce by means of this, the ionization or the radioactive substance. When they enter into the chamber, automatically you can have the digital value. What is the radiation made? A Geiger-Müller counter is a device used for the detection and measurement of all types of radiation, alpha, beta and gamma radiation. Basically, it consists of a pair of electrodes as shown in the figure, surrounded by a gas, usually argon or helium. The electrodes have a high voltage across them. The gas used is usually helium or argon. When radiation enters, when the radiation of your product or the precursor which you are injected or which you have fed into the plant, and isolated compound, what is its biotransformer compound? If the compound containing particular labeled compound, labeled carbon atom or labeled nitrogen, the structure will be changed. That will give the emission of the delayed radiation, which may be alpha, beta or gamma rays, which the radiation enters. Once the radiation enters in the tube, as shown in the picture, it can ionize the gas, that is argon or helium, which is present in the instrument. The ions and the electrons which is produced, ionized compound, they will attracted to the electrodes, the detecting place, and the electric current will be produced. That will be made into an amplifier and gives the digital data. A scanner or a scaler or a scalar, you can say. The scalar counts the current pulses and one obtained a count whenever radiation ionizes the gas. And the next one is scintillation counter. It is little advanced. Scintillation counter will have an metal sodium with a thin film of window where the X-Y rays will be allowed to fall, and there will be a photomultiplier tube which contain an cathode and an amplifying anodes which can give an electrodes of a code of negative and positive leads. High voltage will be applied to increase the multiplication of the small impulses which is obtained from the radiation of your very minimum compound. Very sensitive. Scintillation counter is used to convenient film screen radiography. Many digital radiographic receptors, fluoroscopy, scintillation cameras, most CT scanners, and PET, that means scanner positron emission tomography. Scanners positron emission tomography. Scintillation detectors consist of scintillators and a device such as PMT, photomultiplying tube or photomultiplier, that converts the light energy to an electrical signal. Photons will be converted into electrons, and then they are packets of electrons, photons. They will be converted into electrical signal. And the most sensitive, it is more sensitive than Geiger-Müller counter. Widespread detection. The principle behind the scintillation counter is incident ray interact with the material. Atoms are raised to an excited state. You know very well the ground state will be excited state to any atom, and the excited state emit a visible light like a fluorescence. These photons will be collected by the cathode, and using an anode it will be multiplied, and then you can have a digital display of what is the electricity produced. Light strikes the photosensitivity of a surface of the photomultiplier as shown in the picture. Releases of the proton or the photon electrons, multiplication will take place by means of the anode which is present there. And next you can see that autoradiography, detection of radioactive compounds with a photographic emission like X-ray film. The types in vitro. Autoradiography, receptors are labeled in intact living tissue by systemic administration on the radioligand PAT, positron emission tomography. In vitro autoradiography, slide mounted tissue sections are incubated with radioligand so that receptors are labeled under very controlled conditions. Uses of this autoradiography: map anatomical location of radio-labeled ligand to visualize and quantify receptors in tissues. Tracer neutrons by axonal transport of radioactively labeled amino acids. Certain sugars are transmitter substances. Measure DNA production. Example, H3, the isotope deuterium, H3 thymidine is used. An in vitro autoradiography picture, you can see this scanner. When a radioactive isotope in a ligand decays, it can be measured by positron emission tomography, PET, a single photon emission. An animal also, It's possible for you to take a part in a slide or a section of a tissue, and radioligand equilibrium binding can be identified by a fixing exposure, by phosphor imaging scanning or scan in digital auto radiogram. You can quantification can be done. Mass spectrometry. Mass spectrometry, MS, is the analytical technique that measures the mass to charge ratio of charged particles. It is used to determine masses of the particles. For determining the element composition of a sample or molecule, and for elucidating the chemical structure of molecules, as peptides and other chemical compounds. It will give you daughter ions, and that ions can give you the mass of the particular element. NMR spectrometry, nuclear magnetic resonance, where this spectroscopy, NMR spectroscopy, is a research technique that exploits the magnetic property of certain atomic nuclei or atomic nuclei to determine, it gives that magnetic property to determine the physical and chemical properties of the atom, like carbon and nitrogen, or the molecule in which they are continued. It relies the phenomenon of nuclear magnetic resonance and can provide the detailed information about the structure, dynamics, reaction rate, and the chemical environment of the molecule. Methods in the tracer technique, as we have discussed, again this slide shows. One is about the precursor product sequence. Number 2, double multiple labeling. Number 3, competitive feeding method. And number 4, isotope incorporation. Number 5, sequential analysis. Other techniques, use of isolated organ, grafting methods, and use of mutant strains. In precursor product sequence, in this technique, the presumed precursor of a constituent under investigation on a labeled form is fed into the plant, and after a suitable time, the constituent is isolated. You want to see what is the change has happened. Purified, and radioactivity is determined by Geiger-Müller counter or by means of scintillation counter or any other means. Radioactive isotopes can be identified by mass spectrometry or NMR. So this disadvantage in this method, the radioactivity of the isolated compound which you fed to the plant alone, it is not usually sufficient evidence that because the particular compound fed into the direct precursor, because the substance may enter into the general metabolic pathway, other pathways also, and from there may become randomly distributed through the whole range of a product. So we have to do trial and error, and we have to find out. Application of this precursor product sequence is stopping of hordenine production in barley seedling after 15 to 20 days of germination. They are finding out this one. And restricted synthesis of hyoscine, which is present in Datura stramonium, distinct. What is the distinct from hyoscyamin in Datura stramonium can be identified. This method is applied to the biosynthesis of morphine and ergot alkaloids. And number 2, double and multiple labeling. So double labeling, we know that. So what do you mean by double and multiple labeling? Maybe 2 carbon and nitrogen both together can be labeled and compare. This method is an evidence for the nature of biochemical incorporation of precursor rising double and triple labeling. Even multiple labeling, triple also possible. Carbon, nitrogen, and also if there is any phosphorus or sulfur or sodium present in the compound can also be made. And in this method, specifically labeled precursor and their subsequent degradation of recovered product are more employed. Application: this method is extensively applied to study the biosynthesis of plants, plant secondary metabolite, complication, many big structure used in a study of morphine alkaloid. Double and multiple labeling, a research, an example you can see, Dr. Litty used a double labeled lysine. Lysine is an aliphatic amino acid with carbon 14 a specific position fed to the Nicotiana gluca plant to trace the origin of the piperidine ring, piperidine ring, a 6-membered ring with an NH2, piperidine ring in the alkaloid, that means nicotine, or even in the anabasine, demonstrating with a hydrogen atom from lysine, where incorporating, providing lysine's role in the forming of the ring structure. How it is, lysine is an aliphatic open chain, how it forms. It is forming a ring structure. Piperidine have been decided by means of Dr. Lenti. A classic application of isotopic tracer technique in natural product biosynthetic study he has given. You see this picture, it will give you understanding. You see the star which is made, a carbon atom is labeled carbon 14. A nitrogen atom is also labeled for N15. So, in Nicotiana, the plant, Nicotiana plant, it is inserted gluca. Nicotiana gluca plant, it is converted into, see that particular star, both of them are present in the anabasine. So this is what the study, multiple or double labeling and seeing in the product whether it is present or not. It is present and it is shown. And even we can say the lysine with a single, this is what single, and you are seeing what is the change. Multiple and as well as single or double or even triple can also be done. This is called labeling, multiple labeling compound. In the same compound having two isotopes, making it labeling by two isotopes or three isotopes, radioactive isotopes as shown. Competitive feeding, competitive. Which one is the intermediate product? Which one is competing? There will be, you know about the antagonist. Many antagonists are there. Competitive feeding, competitive antagonist in your pharmacology what you study. So competitive feeding, if incorporating is obtained, it is necessary to consider whether this is fact the normal route of synthesis in plant, not the subsidiary way. Whether this goes through B or it goes to B dash to form C. So you can see that competitive feeding is distinguishing whether B and B dash, the intermediate product, normally intermediate in the formation of C from A. Like A to C, whether it follows the B pathway or B dash pathway. Whether A to C, it passes through the B or B dash can be identified. In application, this method used to elucidate the biosynthesis of propane alkaloids, biosynthesis of hemlock alkaloids, conelyne or conhydrin. These are present in biosynthesis of alkaloids of Conicum maculatum, hemlock. Use the carbon 14 labeled compound as shown in the picture. In lysine, they are seeing that whether lysine is giving rise to conelyne or it is passing through, that is, it is whether competitive feeding or it is poly beta keto acid is giving to conelyne, or so it is the intermediate product. Gamma conysine is responsible to give conhydrin, or lysine passes through conelyne directly, or the poly beta keto acid is responsible, which can be identified by means of having a very clear carbon 14 in poly beta keto acid, or even lysine can also be used for this purpose and to study. So this is how it is how we are labeling the compound and identifying. This is competitive feeding method example. You can get how you can achieve this conhydrin present in the hemlock plant. Conium maculatum example. With example and interesting to know about what is there. An isotope incorporation. This method provides information about the position of the bond cleavage, their formation during reaction. Glucose 1-phosphatase, an enzyme cleavage as catalyzed by alkaline phosphatase. This reaction occurs cleavage of carbon oxygen bond or phosphate oxygen bond. You can see how the phosphate and the oxygen 18 having the finding out the reactions can be seen. When you see sequential analysis, here the principle of this method of this investigation is to grow the plant in atmosphere completely a carbon dioxide with carbon completely with 14 radioactive carbon dioxide. Then analyze the plant given intervals and obtain the sequence of which correlated compound become labeled. So carbon dioxide with carbon 14 or sequential analysis has been very successfully used for in elucidation of carbon in photosynthesis. Determination of sequential formation of opium, hemlock and tobacco alkaloids. Exposure as less as 5 minutes with the atmosphere of carbon 14 carbon dioxide is used in detection or detecting of biosynthetic sequence as pipritone, menthone, menthol in mentha preparata. An application of trace technique when you talk about the study squalene cycle by using squalene cyclization. We have studied this. Squalene is formed from in the acetate pathway is the 30 carbon atom. 2 molecules of farnesyl pyrophosphate can give rise to squalene. A open chain. Cyclization takes place by epoxide. So in that how to form? You can use a trace technique by using carbon 14, a labeled compound of hydrogen 3 labeled mevalonic acid, and how the mevalonic acid which is having carbon 14, if it is present in the squalene, it is confirmed the mevalonic acid is converted into squalene. So intermediate products can be identified. Study of squalene cyclization by use of carbon 14 and 3 hydrogen, that means tritium hydrogen labeled mevalonic acid. Interrelationship among 4-methyl sterols, 4,4-dimethyl sterols, that means terpenols or isoprens are used in carbon 14 acetate. And terpenoids biosynthesis by chloroplast isolated in organic solvent by use of 2 molecules of carbon 14 in mevalonate. You know 6 carbon atoms are there. So you can have study the formation of cinnamic acid in pathway of coumarin from labeled coumarin. So when you see the other applications, number 5, origin of carbon and nitrogen atoms of purine ring system by use of carbon 14 and 14 nitrogen labeled precursor. Study of formation of scopolamine which is present in this in a tropane alkaloid by use of labeled phenylalanine. By use of calcium 45 as a tracer, found that intake of calcium by plants from soil, carbon, and that is calcium oxide and calcium carbonate. And you see, by adding ammonium phosphate labeled with 13 phosphorus, 32, not 13, 32 phosphorus of non-specific activity, the uptake of phosphorus is followed by measuring the radioactivity as labeled research first is low part of plant than the upper part, that is branches and leaves, etc. So other methods used for biosynthetic investigation are isolated organ tissues and cells, like isolating the tissues of the animals or even the plants, can also be used for various in vitro studies. And grafting, especially by means of grafting in vitro study, which is used normal, for example, by shoot and the root. And mutant strains also can be taken as an organisms, or we can use enzymes isolated from the particular plant, can also be used for various studies, can be done by DNA, RNA changes, hybrid varieties can also be studied, and resistance can also be studied by biosynthetic investigation. And you see here isolated organ tissues. This method is based on use of isolated part like plants, stem and root. This technique is useful in determination of site of biosynthesis of particular compounds. Example, roots and leaves of study of Nicotiana and Datura, petal disc of study of rose oil, tropane alkaloids in root of Solanaceae family. And grafting methods, this method is used for the study of alkaloid formation by grafted plants. Example, tomato Sichon grafted on Datura produce alkaloid, while Datura Sichon, the shoot part, grafted on a tomato produces less quantity of alkaloid. So identification, which one produces alkaloid. This shows the main site of alkaloid is biosynthesis in root, not in the shoot. So this is how the grafting of the Sichon wood and the cambium matches, how the straight sharp cambium part should be inserted and it will be locked. There are many supplements which can support for this grafting is available in the market. Use of mutant or mutant strains. Mutant strains, in this mutant strains of microorganism are produced with a lack of certain enzymes. So sometimes the enzyme, control of the enzyme, example, gibberella mutant. Gibberellins, you know that gibberellin auxins, growth hormones, is used to produce isoprenoid compound by means of Lactobacillus acidophilus. Lactobacillus acidophilus is used in mevalonic acid pathway and isoprenoid biosynthesis. Enzymatic study, phytohormones like auxin plays critical role in regulation of the plant growth and development. That is IAA, indole 3-acetic acid, auxin, has been recognized as a major auxin which is synthesized in the plant. Still it is unclear, but previous research, a genetic and the enzymatic study demonstrated that both tryptophan, aminotransferase of Arabidopsis TAA, and YUCAA or YUCA, that is flavin monooxidogenic enzyme like protein, require this biosynthesis of IAA, that is indole acetic acid, auxin, the major auxin during plant development. That is tryptophan aminotransferase of Arabidopsis, that is TAA, family produces indole 3-pyruvic acid, IPA, indole 3-pyruvic acid, and YUC, that is family function of, that is flavin monooxygenase like protein, functions in the conversion of IPA, that is indole pyruvic acid, into indole acetic acid, auxin, in Arabidopsis in the plant Arabidopsis thaliana. You can see this is the Arabidopsis thaliana. Arabidopsis, you can say, thaliana plant. And this is a picture which is just to show you about the various metabolic pathways, basic primary metabolic pathway which rises to even produce fructose, disaccharide, monosaccharide, and also monosaccharides, 5-membered rings, which we have to study to give the basic structures. Basic can be biosynthetic pathway can be studied. Thank you. Once again, just listen, understand, write yourself, make clear, and submit your report. Thank you.